Philipp Mitteroecker.
Oh, come on, we tried so hard to pronounce
I really tried.
We that together.
Yeah. Mitteroecker.
Mitteroecker.
Thank you. Thank you. I needed that. Associate Professor in the Department of Evolutionary Biology's Unit for Theoretical Biology at the University of Vienna in Austria. Philipp is the theoretical biologist and evolutionary biologist with strong interests in human and animal evolution, evolutionary medicine, biological anthropology, statistics, and philosophy of science. Second speaker, Barbara Fischer, Elise Richter Fellow in the same unit at the University of Vienna. She is an evolutionary biologist and data scientist who studies phenotypic evolution using theoretical and empirical approaches, specifically focused on the classical problem of adaptation versus constraint in evolution. And they will be presenting today on why is human childbirth so difficult, obstetrics, and the evolution of labor. So take it away. Over to you, Philipp.
Thanks a lot. Thanks again for inviting us to this fascinating club here. I know several of the faces and several of the names here that I see, so also hello to you. Okay, I try to share my screen now. Does it work?
Yes.
Yeah. Okay,
yeah.
So let me make that full screen. Okay, so Barbara and I will talk about human childbirth today because it's an interesting evolutionary puzzle and a pressing health issue at the same time. And we worked on that a lot in recent years. Viviparity or live birth has evolved not only in mammals, but also multiple times in other vertebrates, including sharks, amphibians, and lizards. Viviparity arose back in the Jurassic, and the true placentals emerged in the Cretaceous more or less 90 million years ago. So one should think that childbirth in human should really work because there was enough time for evolution to make it work. Nonetheless, you all know that that's not really the case. That there's a surprisingly high rate of obstructed labor, which means that the baby does not exit the pelvis because it's physically blocked despite normal uterus contraction. There are multiple reasons for that. The reasons we want to focus on here is the anatomical disproportion between the fetus and the maternal birth canal, or feto-pelvic disproportion. And the main reason of that is that the head of the baby is too large, or cephalopelvic disproportion. The actual rates of cephalopelvic disproportion or feto-pelvic disproportion are very difficult to estimate. Many of you will know because in the global north, at least, usually the gynecologists don't try to the very last when it becomes dangerous. Of course, they do a C-section earlier, so it's not easy to find out whether baby would have passed or not. And of course, that's not in practice, that's not always a yes or no thing, that can be a matter of degree. Also, when a C-section is done, mostly it's reported that it's because of cephalopelvic disproportion to make the health system pay for it. So it's not easy actually to get the proper numbers of that. But for example, in Africa, rates ranging from 1.4 to more than 8% have been reported. A large US study reported 2.3% rate of cephalopelvic disproportion for infants weighing 3 to 4 kilograms and 5.8% for infants weighing more than 4 kilograms. So it's still considerable rate. From an evolutionary perspective, this translates into strong selective pressure because without proper obstetrics and medical treatment and often a cesarean section, that can be lethal to both the mother and the fetus or lead to severe morbidity. Why is human childbirth so risky? Well, the obvious reason is that compared to other great apes, the human fetus is so large compared to the birth canal. But why is that the case? Well, humans evolved, or in our lineage, the humans evolved bipedal locomotion more or less 6 million years ago, and only in the last 1 or 2 million years, the human brain started to become so large. So basically, when with the evolution of bipedal locomotion, also the pelvis changed a lot in form and became narrower. And then when the brain increased in size during human evolution, basically we were stuck with this pelvis. Well, so far so good, but this raises the question, why did the pelvis then not evolve to be wider again, right? Because in principle, we have everything that's needed for evolution. We have heritable variation and we have selection. It should have worked. It should have become wider, basically. And already in 1960, Washburn suggested a sort of trade-off situation, that he suggested that a wider pelvis would be energetically disadvantageous for locomotion. So in a real physical talk, I would not try to walk like a duck to simulate how basically if the legs are too far apart, how silly that would be and how inefficient I would walk. I don't want to make a fool of myself here online, so I don't try that. But you can imagine that even though that sounds quite reasonable and probably there's something behind, it has only been tried to be tested in recent years, but these studies didn't really support that. So it's a bit unclear, even though there's probably something behind it. There's further work is needed to show that really. Another factor, and Barbara and I believe, and also others, that this is an important factor, is the pelvic floor. In upright humans, the weight of the inner organs and the fetus must be supported by the pelvic floor, unlike in quadruped primates where this is not the case. And this is an idea that goes back to Abitbol. And he argued that the pelvic floor muscles evolved not for that purpose, but mainly for continence and controlling the tail. And now in humans, where we are upright, it has to have this function. And it's basically a vulnerable part. And so a wider pelvis would make the pelvic floor even more vulnerable. And so there is sort of a trade-off between having a sufficiently large pelvic canal, a birth canal, and a supportive pelvis for the pelvic floor. This is supported by some, but not all, clinical studies in this field. So some studies reported that women with a larger pelvic canal indeed have a considerably increased risk of all kinds of pelvic floor disorders like prolapse and incontinence. And also this year we published a study together with Katja Stansfield, Krishna Kumar, and Nicole Grunster. I've seen that Katja is also here. Hello, Katja. So here, Katja, she's an expert on finite element modeling. And what she did is she modeled the pelvic floor using a finite element model. This is what engineers use to simulate objects under pressure, like a bridge or parts of a car or things like that, and see where they are likely to break and where they deform and how strong the strains and stresses are in the material. And so what we did is we had this pelvic floor, and we basically constructed pelvic floors of different sizes, basically corresponding to differently sized birth canals. And we found that indeed that the larger the birth canal, the larger the pelvic floor, the more strongly it descends, and the higher are the strains and stresses in the pelvic floor tissue. So also we interpreted this as support of this pelvic floor hypothesis. And pelvic floor disorders are relatively common in humans. There is an up to 40% prevalence of uterine prolapse among US women, and prevalences of urinary incontinence in the order of 20 to 50%. Of course, clearly this increases with age and with the number of children and the mode of delivery and so on, but also in young women that's present. So, and of course, today nobody dies of that, luckily, well, at least in the global north, but without medical treatment, prolapse and chronic incontinence poses a considerable risk for urinary tract infections and can lead to social ostracization, which is unfortunately pretty well known from sub-Saharan Africa, where this is quite an issue. And of course, this in an evolutionary context, again, lowers fitness. So to sum that part up, the human pelvis obviously is a compromise solution, trading off birth canal size against other pelvic functions, including pelvic floor support, probably also locomotion and other issues that I don't touch here. But what I always found kind of puzzling is how can the considerable mortality without medical treatment resulting from obstructed labor be outweighed by presumably, presumably small advantages for locomotion and pelvic floor stability. How can one imagine that? For me, this was always kind of a bit counterintuitive. And so in a 2016 paper with my colleagues Simon Hutecker and Barbara Fischer and Michaela Pavlitsev, we tried to model that and see how that would actually work. And we call this the cliff edge model for reasons that will become obvious in a minute. And this model starts with a pretty gross and bold simplification. We are, of course, aware of that, but I'll talk about that later, and I'm happy to discuss about that after the talk. We simplified that all to a single variable, which I call D here, the difference between the size of the neonate and the size of the maternal pelvic canal. In the sense that if this hypothetical variable D is larger than 0, this would indicate that the fetus is larger than the birth canal and it wouldn't fit through. So we have the case of feto-pelvic disproportion. A D smaller than 0 would indicate the fetus is smaller than the birth canal, and in principle, it would pass through if nothing else happens, right? So what do we know about selection on this proportion, on this feto-pelvic proportion or disproportion? Well, we know from many studies that large newborns have less risk for all kinds of diseases and higher survival rates, which translates into positive selection for a large value of D. And we know, for example, from these pelvic floor studies that I mentioned, that a narrow birth canal, so a small value of D, a small pelvic canal would be favored for these reasons. So what this means is that there is probably selection for a large fetus and a small pelvic canal, so basically for a large D value. And we can visualize that. So here I plotted, I don't know, by the way, do you see my cursor here moving around? No?
No.
Okay, then I stopped doing that. There is this, so I plotted this D variable here against fitness, individual fitness, so average reproductive success. And this data suggests that the larger this value D, the higher the fitness, because the baby is larger, the birth canal is narrower, the pelvic floor is supported, well, until to the point when the baby doesn't fit through anymore. Then fitness drops suddenly because, again, without cesarean section, and so that's probably lethal, and so fitness drops suddenly. The data we found indeed supports that this increase is roughly linear, but of course it isn't exactly. So this is an idealization, right? And also, of course, the drop is not that strong, but it kind of looks like this. And we call this a cliff-edged fitness function because it looks a bit like a cliff that and you can follow from that. The problem is that the phenotype doesn't look like that. The phenotypic distribution is pretty like a normal distribution here. So there are many data on pelvic dimensions and neonatal dimensions, and they're all more or less symmetric. So we have a sort of symmetric phenotypic distribution and a highly asymmetric fitness distribution or fitness function. And it's pretty obvious that this can't work out well. So what we see here is that given that the fitness increases with a larger D value, that this phenotype distribution will evolve to the right. So it will evolve towards larger D, towards larger newborns and smaller pelvises, until some of the individuals fall off the fitness cliffs, right? So this shaded area corresponds to the incidence of feto-pelvic disproportion, to the number or to the rate of mothers that experience feto-pelvic disproportion. And with a bit of math, one can actually calculate where the evolutionary equilibrium would be. So basically, what would be the optimal, evolutionary optimal, not individual optimal, the evolutionary optimum for this trade-off? Because I don't go into the details here, but you may know what evolutionary theory tells us is that what's maximized by evolution, by natural selection, is the average population fitness, so the average reproductive success. And we can actually calculate that optimum here. And in this particular case, for this fitness increase that I've drawn here, and this width here, that turns out to be 4.5%, the rate of feto-pelvic disproportion. And in that case, then evolutionary stops, because then it had reached this sort of optimum. Well, what influences that rate? Clearly, the steeper the fitness increase, the higher the rate of feto-pelvic disproportion, because the steeper this blue curve, the more it pushes the rate distribution towards the fitness cliff. Also, it's pretty clear that if the phenotype distribution would be narrower, few individuals would, less individuals would experience feto-pelvic disproportion, right? The tail towards the fitness or above the fitness cliff would be shorter. But it turns out it's not easy to achieve that evolutionarily to reduce this variance of this value D of the proportion between maternal and fetal dimensions. Why so? Because this D, this proportion is actually influenced by 3 genomes distributed over 2 generations. Because there's the maternal genome influencing the maternal dimensions, and there's the fetal genome, half of which is paternal. And so we have many genes here, and the neonatal genes inherited by the father are uncorrelated with those of the mother. It means that these variances add up here into this in this ratio. So the variance of this proportion between maternal and fetal dimensions are higher than the variance of maternal and fetal dimensions separately. And also, of course, we have pretty strong environmental influences here. We know that nutrition and so on affects not only the maternal pelvic dimension, but also clearly the neonatal dimensions. Gestational age is highly plastic. We know that pelvic form changes over age, also in adults. So we have a lot of environmental components here too. All that means that the variance of D cannot easily reduced by stabilizing selection. So can we apply this model to real human obstetric data? And it turns out, yes. So if we assume that this linear fitness increase is close to linear and the distribution approximately normal, the system turns out to be mathematically determined by a single parameter. And that's the fitness increase per standard deviation of D, which is called the standardized selection gradient in evolutionary theory. And so what we can do is basically, before I've shown you that if you would play around with this increase of the fitness, so the steepness of the blue curve, you would get different rates of fetal-pelvic disproportion. Now we do the opposite. We have rough estimates of the actual rates of fetal-pelvic disproportion, and we see which fitness increase would produce that. And so I told you these disproportion rates are not easy to estimate. So we take here a range of something in between 2 to 6%. And it turns out that this translates into standardized selection gradients of 0.06 to 0.016. Well, is that a lot or not? Luckily, there are many studies in evolutionary biology where researchers have tried to estimate selection gradients for all kinds of animal and plant traits. And there are some very comprehensive review papers, and they report median or mean selection gradient in the order of 0.15 or 0.2. So this tells us that the strength of selection towards a large baby or a narrow birth canal is not that strong, or does not need to be that strong in order to produce that relatively high rates of fetal-pelvic disproportion. So this is basic. For me, this was counterintuitive, because I had expected the opposite. I would have expected very strong selection necessary to maintain this birth problems, basically, but it's not true. Because of this cliff edge property, a relatively weak selection is sufficient to produce these rates of fetal-pelvic disproportion. And I have to admit that men make it all worse, because so far I have only modeled female traits. But of course, there are not only females, there are also men. There are also males. And so if I make it a bit extreme now, so we have a sex ratio of 1/2. And if we assume that males have no fitness loss due to fetal-pelvic disproportion, because, well, actually, Barbara said I should never phrase it like this. Sorry, Barbara, for doing it nonetheless. So if the father basically would not suffer a fitness loss because the mother and the baby dies because he would reproduce with somebody else, then it would be like this. Of course, in practice, that's not like this. It's somewhere in between. But in this extreme case, we can imagine that for males, sort of the fitness increases with a larger D value, but for females, it drops to 0. So overall in the population, it would look like this. And what this means is that because the genes that control pelvic dimensions and fetal dimensions are expressed and inherited by both sexes, but only under obstetric selection in females, not in males, the situation is aggravated. So this leads to an even higher rate of fetal-pelvic disproportion. So I call this here model 2. I mean, the reality is certainly somewhere in between, right? And certainly also population, it depends on population structure and culture and obstetric practices and and many factors. And also differs between populations, therefore, and cultures. And so under this model, it needs even less selection towards a large baby or a narrow birth canal. So here we would get selection gradients in the order of 0.02 to 0.07. So this is really weak selection necessary to achieve the observed rates of fetal-pelvic disproportion. Okay, what about cesarean sections? Luckily, in many countries, there is cesarean section available. What does this mean in this kind of cliff edge model context? So you see here the model, the cesarean section removes the cliff edge. So with cesarean section, everybody survives a large baby or a narrow birth canal. And what this means clearly is that the previous equilibrium is disrupted and evolution starts anew. The distribution is expected to move to the right. And given that we have already achieved rough estimates of the selection gradients, and by no means we pretend that this is exact, because of the many assumptions, the many simplifications there, this is just meant to be a rough order of magnitude. Given that we have some estimates of heritability, and if we assume now as a starting point a 3% incidence of fetal-pelvic disproportion, model 1 suggests that when cesarean section became regularly available, and that, at least in Europe, and I guess also in the US, was some point after the Second World War, so like in the 1950s or so. Of course, it was available also before, but that it was really available and frequently available and a safe procedure. That was roughly like 2 or 3 generations ago from now on. And we estimated that in this time, basically, so we took here 2 generations, the incidence of fetal-pelvic disproportion would have increased by something like 20% in model 1 or roughly 10% in model 2. So this would be about half a percentage point, so like from 3 to 3.5%. Again, this is by no means exact, just as a rough order of magnitude. Given that we have C-section rates of 30% and more, this is of course not much, this half percentage point. Nonetheless, it's intriguing that we expect here to see an evolutionary change triggered by medical intervention within only a few decades. And of course, one can ask and should ask whether that's true. And it turns out it's not easy to recognize in the epidemiological data, but we successfully validated the model by predicting the inheritance of C-section. But I don't go into that. I would like to conclude by my part by mentioning that, again, that medical and technological advancements do not necessarily reduce evolutionary change. Clearly, they reduce selection, but because many traits in humans evolved not by ongoing directional selection, but by trade-offs, they evolved as compromises to a complex situation. If in such a trade-off, one direction of selection has been removed or lowered by medical advancements or technological advancements, something remains and can lead to a new evolutionary change. It disrupts a previous equilibrium. And this can change human health and biology, not only within thousands of years, as many people, at least in the public, expect, but actually within a few decades. And I think it is important to study and understand these complex biosocial dynamics to guide public health and research politics. And I'd like to emphasize that this is not, of course, not only a biological system, it's really a biosocial system where many social and cultural phenomena influence that too. And I guess this will be also part of the ongoing discussion. My part is over now. And I think Barbara will start now, unless there are some pressing questions.
Yes, thank you, Philipp. Then I'll take it from here. Let me see if I can share my screen. Yes. Oh, come on. This is not the start. This is where we start. So thanks for the nice introduction, Philipp. And so I can skip some of my own introduction because you all know so much already about this evolutionary conflict that humans and especially human females are subject to. I want to talk a little bit now about the evolution of sex differences in the human pelvis. This is some work we recently published together with Nicole Grunstra, Eva Zaffarini, and Philipp and me. And I also saw that Nicole is here, so hi, Nicole. Glad to have our team members support us here tonight. It's actually evening in Austria, so I can say that. The human pelvis is the most sexually dimorphic region of the human skeleton. It's actually the pelvis. The pelvis has dimensions where females are larger in absolute terms than males. So not just relatively, if you scale it, if you consider the dimensions relative to body size, but they're actually absolutely larger. So there is something about obstetric selection that plays an important role here in shaping our morphologies. And this is also something forensic scientists make use of, for example, when they try to identify the sex of skeletons. They look at the pelvis. But we know very little when this sex difference is actually emerged in the evolution of modern humans. this is because the fossil record is so patchy. So here I'm showing you the famous skeleton of Australopithecus Lucy. And at some point, there was actually a debate in the literature whether Lucy was actually female. So it's of course hard to identify the sex of an individual if you have nothing to compare it with, right? How can you tell whether this individual was female or male if you have one only? I should also say that by now this debate is considered somewhat settled. So the mainstream opinion is that Lucy was, of course, female, but often things are not that clear. Now, when it comes to Neanderthals, for example, I'm showing you the best preserved Neanderthal pelvis specimens that we have. On the left here is a reconstruction of the Taboon pelvis, and on the right is a very recent one that has only been published a few months ago by Adekbodeba et al. Of the Kebara 2 pelvis. And so that's what we have. So this is what we know about sex differences in the pelvis in Neanderthals, and it's very little. I mean, these are very sophisticated reconstructions, but nevertheless, of course, there's a lot of room for interpretation. People base interpretations of birth in Neanderthals on those individual reconstructions of individual pelvises. So this is, of course, speculative to a certain extent. So what I want to present now is our recent study, namely a comparative study. So we looked at humans and chimpanzees and compared the two species. And we did that by a morphometric approach. So we looked at the number of 99 humans from the Haim and Todd skeletal collection and the number of chimpanzees from a number of online repositories in the Natural History Museum in Vienna. And we landmarked them. So we used landmarks, homologous points on the pelvises of both species. And I'm gonna show you the results of this analysis on the next slide. So what I'm showing you here are the average pelvis shapes in humans in the top row and in chimpanzees in the bottom row. And here in the middle are the average female and male shapes, and to the left, very left and very right, you see extrapolations. So these are produced simply by taking the vector that describes the sex differences between females and males in the respective species and just adding it on top once again. So adding the sex difference vector on top of the average male pelvis shape then produces this ultra male shape, and this, by subtracting it, I then get the ultra female shape. And you're probably familiar with those features that are actually sexually dimorphic in the human pelvis. It's the you cannot see my cursor, can you? You can?
We can see yours.
Very good. So I can actually point at things. So the subpubic angle here is wider in females than in males, and you see this particularly clearly if you look at the exaggerated shapes here to the very left and very right. This is one of the features that is well known to be sexually dimorphic in humans. Also, the sacrum protrudes deeper, is longer in males than in females, and protrudes deeper into the pelvic canal. And all of these dimensions that are actually relevant for obstetrics in the pelvic inlet and in the entire pelvic canal are larger in females than in males. Particularly if you look here, the transverse diameter up here in the pelvic inlet is relatively larger in females than in males. In fact, it's also absolutely larger, but here we're talking about shape. So this is about relative differences, which is particularly clear if you look here at the exaggerated shapes. So these are the patterns we know. These are the patterns forensic anthropologists use to sex skeletons. Now, if we look at the bottom row and compare the chimpanzee pelvises, those patterns seem to be there as well. So if you look at the subpubic angle here with me, it's not so clear if you compare just the average female with the average male shape. But if you look at the extrapolations, it's clear. So also here we have a much larger subpubic angle in the ultra female than over here in the ultra male shape. Also, if you compare the relative length of the sacrum is relatively longer in males clearly than it is in females. So again, this is the same feature that we have in humans. And also, if you look at this, the width of the pelvic inlet, the transverse diameter here, there's just more space in the pelvic canal in females than there is in males. So it seems that chimpanzees, although they have much easier births than humans, because the neonate is relatively smaller compared to the dimensions of the pelvic canal in chimpanzees than in humans, they have the same features of sex differences. So now let me show you another analysis here. We then did a principal component analysis of this shape data. And what I'm showing you here is are two panels that depict shape space. So each of these points here is an individual. The blue points are the males and the red points are the females. And those big dots in the middle are just the sex mean. So this is the average human pelvis shape, the average male human pelvis shape, and this is the red one is the average female. And then over here, the light blue and pink data clouds correspond to the chimpanzees. Again, the blue ones are the males and the pink ones are the females. And what you can see immediately here in the panel on the left is that the first principal component separates the species. Humans end up land here on the left and the chimpanzees land here on the right. That's not very surprising, right? The species differences were huge as you've seen on the last slide. But what if we take those out? If we look if we take out the species differences, then that's what I'm showing you here on the in the second plot, where principal components 2 and 3 are plotted on the axis. And what you can see here is that the sex means of the chimpanzees, the pink big dot and the light blue big dot, they fall right in between the sex means of the humans. So the sex difference between in chimpanzees, the difference in terms of shape of the pelvis in chimpanzees, when the species differences are taken out, are aligned with the sex differences in humans. The sex difference vector points almost in the same direction. Now, those little black vectors that I've plotted here, those depict the allometry. So those I've only plotted here to show that this is actually not allometry. These are not consequences of size that we're looking at here, but a sex difference is actually clearly distinct from size. So it seems that the patterns of sex differences indeed are aligned in the two species and that they are almost identical. Now, in another analysis, I've done something slightly different. Now you already know how to read this plot. So the first row here is again just the human sex difference, again extrapolated on the very left and very right. But what I've done here in the bottom row is that I took the vector of sex differences from the chimpanzee data alone, only calculated from the chimpanzee data. And I took this vector and added it on top of the human sex means to produce these extrapolated shapes here in the bottom row. So the pattern that is shown here in the bottom row is the human means, the sex difference vector from chimpanzees added on top of the human pelvis shapes. And you see that the top row looks almost the same as the bottom row, right? So this is just, again, supporting the finding from the previous slide that the pattern of sex differences are the same in the two species. Now, I said the pattern is the same, but the magnitude is not the same. I've shown you previously that the chimpanzee sex means they fall right in between the human sex means, and the vector of sex differences is also much shorter in chimpanzees than in humans, actually by a factor of 2. So the pattern is the same in the two species, but the magnitude is twice as large in humans compared to chimpanzees. But it's pretty obvious because the pattern is so very similar that this pattern did not evolve de novo in modern humans, but it must have been there in the common ancestor of humans and chimpanzees, which means that it must also have been there in Australopithecines and Neanderthals and our extinct common ancestors. But in fact, we think that the pattern is much older. We think that it is of mammalian origin, or maybe it is even older than that. And Let me explain why we think that. Neonatal size varies a lot across mammals, and humans are actually not special in giving birth to extremely large, to very large neonates. There are groups of mammals which give birth to even larger neonates than humans. Have a look at the plot here at the bottom, where we are showing relative neonatal body mass as a percentage of the maternal body mass. Sorry for this. And on the horizontal axis, there's female body mass. And we are here, and here are the other great apes. And have a look at the bats. That's quite crazy. Bats give birth to neonates that have 40% to up to 50% almost of the mass of the mother. Apart from the fact that this is quite insane in terms of metabolism, it's possible. They fit through. So evolution has produced a highly sexually dimorphic pelvis in these species to be able to actually birth these enormous neonates. And this is true not only for bats, but mammal species with relatively large neonates tend to have more sexually dimorphic pelvis. So this is not something that is uniquely human. Also, sex differences in the pelvis have actually been documented for all the major placental clades. And at least qualitatively, from looking at these bones and from what's been described in the literature, these patterns seem to be similar to the human pattern, with the differences mainly concentrated in the pubic region. And then there are some mammal species like this marsupial here, the Virginia opossum, which give birth to tiny neonates. So there's clearly no relevant obstetric selection in the game here, but they still have sexually dimorphic pelvis. The sexual differences here are subtle in this species. They are not particularly pronounced, but they are there. So we actually think that a sexually dimorphic pelvis could be the ancestral condition here among mammals, or maybe it's even older, maybe even among certain amniotes. And we propose a hypothesis, the facilitated variation hypothesis, as a possible explanation for this. A concept of facilitated variation was proposed by Gerhardt and Kirschner in 2007. And they proposed that evolution uses certain conserved core components and recombines them. And in this way, despite the fact that certain parts remain very constant and very conserved, manages to produce high evolvability of certain other traits. And what we propose is that the underlying genetic and developmental machinery that underlies these patterns of sex differences in humans and mammals are such conserved core components, that they stayed relatively conserved during mammalian and maybe even amniote evolution, but that there is a regulatory switch that determines the magnitude of these differences. And the switch, by contrast, has high evolvability. And we think that it might relate to estrogen and relaxin secretion, or maybe to the reactivity or density of the corresponding hormonal receptors in the pelvic tissues, so that evolution actually had found a rather easy path towards blowing up the magnitude while keeping the pattern very constant. And this enabled humans, this basically facilitated pelvic evolution in humans to this degree that we see today. So humans didn't have to evolve pelvic sex differences from scratch. They were already there. So obstetric selection could just use those pre-existing patterns and blow them up. That's what we think has possibly happened. And now I want to thank you for listening, and I'm very curious to hear your questions. And yes, I also put our contacts here in case we do not manage to get through all of the questions, or in case you come up with an important question after today's session.
Great, thank you so much. That was a wonderful presentation. So we have a ton of great questions in the chat, and feel free to virtually raise your hand. We do like to hear from people. You can do that by clicking reactions at the bottom of your screen and then raise hand. But to kick us off, we'll go to Jillian Bentley first. Jillian, go ahead with your question.
Yeah, thank you. That was really interesting from both of you. Thanks a lot. One of the things that I wanted to ask, though, was about your measurements of the pelvis, because as I understand it, you were using the Heyman Todd collection, which I believe was mostly poor people that were, unclaimed, bodies were unclaimed, and so they made their way into this anatomical collection. And I'm wondering, because these people then presumably were of lower socioeconomic status, what that might say about their pelvic size and shape, because if they were, say, poorly nourished people from poorer families, the women here in particular, then that might be skewing some of your measurements of these pelvises from this collection. And then that would have a knock-on effect for some of your other calculations. So I'm wondering if you could address that.
Yes, so most certainly this is a very specific sample. It's a museum sample, and in a way, of course, we work with what we have, but those patterns of sex differences that we identified in the human sample, they are so very well illustrated in the literature. They are so persistent across human populations that I'm very certain that there is no immediate bias there. So it's true that the average height, for example, is a few centimeters short, but the patterns of sex differences and the shape variation that we identified in this sample fits with other population samples from other human populations. So I'm not really worried about this biasing the results.
Great.
Thank you.
So it looks like we next have a question from Reinmar Hager. Reinmar.
Hi, thanks. I'm just switching on my video so you can actually see that I really do exist. Hi, thanks Barbara, and thanks Philip for a fascinating talk, which is really far outside my area of expertise, so my question might seem very naive. So why can neonates not have smaller heads? Or in other words, why do they not come out sooner? Or we shift brain development rather to a later stage so that the head is basically smaller when it comes out?
Well, this is a very valid question. And there are many medical studies showing that the sort of the time, also the gestation lengths and also the size of the neonate are under selection. So basically that the later you are born and the larger you are born, the safer you are. And also there are, I mean, you know that, I mean, preterm babies have a lot of risks and also can suffer from cognitive deficits and everything. Louis is here. He's an expert on preterm babies. He can tell a lot about that. So we and also compared to other primates, human, and Barbara has shown that humans are already born relatively immature. I mean, that's not true for the whole groups of mammals, but still, obviously, that's not a viable solution. So that it seems that the selection is too strong on that this could have happened. But also, I mean, overall, these trade-off dynamics certainly include that too. So there are really many factors contributing that, and that have led to this sort of, for some, very problematic compromise solution.
Thanks.
Great, thank you for that. So something that I'm interested in, there's an interesting conversation in the chat about whether the different selection pressures for females and males represent sexual antagonism or parent-offspring conflict. I know you addressed it a little bit in the chat, but I'm wondering if you can speak more about that and perhaps bring in some of the other issues that were mentioned in the review article, like the erectile dysfunction in the review paper.
Yeah. So
you can talk about the erectile dysfunction, Philipp. I can talk about that. Parent-offspring conflict. I think it's both, Meredith. It's both in a way a parent-offspring conflict and antagonistic selection on the sexes. It clearly, so that the fact that we have these sex differences in humans implies that there must be different selection pressures acting on females and males. So if obstetric selection was the only relevant selection pressure in the game here, then male pelvises should just look like female pelvises. The fact that we have sex differences means that there is another selection pressure here. Now, Philipp has mentioned our pelvic floor hypothesis, which seems to become more and more convincing. But of course, there's also the traditional ideas of Washburn, that this is maybe bipedalism alone. Maybe there's something to do with the surface to volume ratios and with the thermoregulation. This has also been proposed. So this is an open question, and which ongoing research tries to deal with. But there must be the identity of the selection pressure has not been clarified ultimately, but the fact that it must exist, otherwise we wouldn't see these sex differences.
Yeah, and I would argue that it could easily be both. And in fact, in my comments, I was not discounting that this was parent-offspring conflict, because I think it clearly is, but it could be driven by asexual antagonism.
Mm-hmm.
Maybe, yeah. I mean, most of these selection pressures are certainly shared among the sexes, because both the father and the mother is interested in evolutionary interested in a surviving offspring, right? But indeed, so there is, I agreed to the comments in the chat that there is a parent-offspring conflict going on, because clearly the fetus wants to be as large as possible, but the mother wants some limits here. So this is a conflict. I mean, you mentioned the erectile dysfunction thing. That was an idea of Michaela Pavlitsev, a co-author of some of our papers, and she suggested that the pelvic floor also has a function in males. I mean, clearly, pelvic floor is important for continence, but also she suggested that it has a role for erectile function in males too. There's surprisingly little data about that, except some studies on pelvic floor training and things like that. But this was an idea, and we are so far we couldn't prove that or assess it in any way. But it was more like to put that forward as an idea. But certainly it's pelvic floor is certainly more important in women than in men here.
I just wanted to say something about the sex difference issue. So Adolf Schultz showed that there was a relationship across primate species in the relative size of the infant head and the amount of sexual dimorphism in the pelvis, right? So anyway, I just thought that was worth mentioning.
Yes.
Yes, thank you. It is indeed, yeah.
Yes, so this was also something that I didn't want to, with the Virginia opossum example, I didn't mean to say that the presence of sex differences across the great apes and across primates is not evidence for obstetric selection. By the contrary, actually, I think very pronounced sex differences are very much evidence for strong obstetric selection. But the fact that evolution hasn't completely removed it in other species hints at it maybe being a vestigial trait in marsupials, for example. And there it's not very pronounced, it's subtle there. But if it is indeed a trait that was present in the common ancestor, that's what we would expect, right? We would expect that there were some remnants somewhere.
Absolutely.
Great, thank you for that. We now have a question from David Haig. David, you can go ahead with your question.
Sorry, it's unfair. It's not a question. It's a comment on this whole parent-offspring conflict versus sexual antagonism discussion. So the first thing to say that selection in fetuses is occurring on both male and female fetuses. So it's not a male-female difference that you're looking at. The second thing is that parent-offspring conflict, if you get down into the weeds genetically, there is a prisoner's dilemma going on how much to demand from mothers, and there's competition between different alleles that are transmitted to offspring. And we know that in a prisoner's dilemma, you're not maximizing fitness. And so I think the work is really, really good, but so much discussion of optimal life histories is assuming that there's one fitness being maximized. And if you have parent-offspring conflict in this system, there is nothing that is being maximized in that sort of sense. You're in the realm of not of engineering maximization, but of game theories and costs of failures of cooperation.
This is a very interesting comment. Thank you. So there's certainly, when we developed this model, I was aware that the situation is more complex than that. So we cut through all that pretty boldly here, that and how clearly this sort of you can translate this in this sort of cliff edge, that the strength of selection differs between the mother and the fetus, for example, here. So that could be considered as an average, but I had always hoped that with some help by somebody who can do it better than me, we can actually extend that model, but so far it didn't come to be true. I hope it will become true at some point.
Great. Well, on that note, it looks like we are at the top of the hour. So I want to thank both Philipp and Barbara again for an excellent presentation and excellent discussion. And before we all tune out, I want to quickly share our upcoming events for summer. So on our Club EvMed website here, we have the excellent talk we just heard. And then our next talk will be on Monday, June 21st, and that'll be with David McAdams of Duke University, talking about evolution management from a game theory perspective and applying that specifically to antimicrobial resistance. That should be a great discussion as well. And then if you keep scrolling, you'll see our other speakers, and we'll have more details about those talks to be shared very soon. So with that, thank you again, Philipp and Barbara, and thanks to all of you for attending. And we hope to see you at the next event. Thanks so much.
Thank you. Thank you all for
again.
attending.
Yeah.